Assalamu alaikum. I, Dr. Mohammad Akram, Department of Eastern Medicine, Government College, University, Faisalabad. I will discuss carnitine shuttle. The activated cytosolic fatty acid, fatty acyl CoA, must be transported to the mitochondria. The mitochondrial membrane is impermeable to coenzyme A. There is a specialized carrier that transport a acyl group from the cytosol into the mitochondrial matrix known as carnitine shuttle. First, an acyl group is transferred from the cytosolic coenzyme A to carnitine by carnitine acyl transferase 1 forming O-acyl carnitine. Second, the acyl carnitine group is transported across the membrane to the mitochondrial matrix. In the mitochondrial matrix, acyl group is transferred to another molecule of coenzyme A by carnitine acyl transferase 2 and carnitine is set free. This free carnitine is now available again to transport more acyl group or fatty acids from cytosol to mitochondria. Carnitine shuttle is inhibited by malonyl CoA, which inhibits carnitine acyl transferase 1, thus blocking the entry of cytosolic acyl group into the mitochondria. The result is the inability to transport fatty acids into the mitochondria for degradation and production of energy. A congenital absence of carnitine acyl transferase in skeletal muscles or low concentration of carnitine due to defective synthesis causes myoglobinemia and weakness after exercise. Total energy produced and used in beta oxidation. Examples: palmitic acid, that is the 16 carbon fatty acids. Complete oxidation of palmitic acid produces 7 FADH2, 7 NADH2, and 8 acyl CoA. 1 ATP is used for the activation of palmitic acid before the start of beta oxidation. 1 FADH2 produces 2 ATP, therefore 7 FADH2 produces 14 ATPs. 1 NADH2 produces 3 ATP, therefore 7 NADH2 produces 21 ATPs. 1 acetyl CoA produces 12 ATPs in tissue cycle, therefore acetyl CoA produces 96 ATPs. Net gain of ATPs. From the complete oxidation of palmitic acid to carbon dioxide and water, including tissue cycles, produces a total of 131 ATPs. 1 ATP is utilized during the activation of palmitic acid. The net gain of ATP is therefore 130. Beta oxidation of other fatty acids. For example, stearic acid, 18 carbon fatty acids. Stearic acid, which is an 18 carbon fatty acid, produces half of 19, 9 acyl CoA. 9 minus 1 is equal to 8 FADH2 and 8 NADH2. 1 ATP is utilized in the activation of stearic acid, therefore the net gain of ATP is 147. Beta oxidation of O chain saturated fatty acid. These acids repeat the four-step process producing acyl CoA until the last cleavage when the three remaining carbons are released as propanyl CoA. Propanyl CoA can be converted to glucose. Beta oxidation of unsaturated fatty acids. In addition to the four enzymes catalyzing the four steps in beta oxidation, these acids require additional enzymes. Beta oxidation proceeds normally until it reaches double bond. The additional enzymes remove the double bond and the normal steps of beta oxidation can then proceed. Ketogenesis. It is the synthesis of ketone bodies. They are synthesized mainly in the liver mitochondria. There are three ketone bodies: acetone, acetic acid, and beta hydroxybutyrate. Acetone is volatile and is excreted in breath. The remaining two ketone bodies, acetoacetate and beta hydroxybutyrate, serves as reserve fuel in the body except liver. Liver cannot utilize ketone bodies because it lacks the enzymes thiotransferase which activates acetylcholine. Ketone bodies are used as fuel by tissues such as muscles, kidney, adrenal glands, and brain. During starvation, after about 3 to 5 days of fasting, brain also utilizes ketone bodies for energy purpose. In patients suffering from diabetes mellitus and during starvation, there is increased breakdown of fatty acid to produce energy. Excess of acetylcholine due to excessive breakdown of fatty acid results in excessive formation of ketone bodies. Acetone is excreted in breath. Acetone breath, acetoacetate, and beta hydroxybutyrate are excreted in urine. Reactions of ketogenesis. Two molecules of acetyl-CoA unite to form acetyl-CoA. Acetyl-CoA reacts with acetyl-CoA and forms HMG-CoA, which is then cleaved to form acetoacetate, that is one ketone body. HMG-CoA reductase is a rate-limiting enzyme and is present in significant quantities only in liver. Acetate may be reduced by NADH2 to beta-hydroxybutyric acid second ketone bodies. Acetate undergo non-enzymatic decarboxylation to form acetone, the third ketone bodies. Acetoacetyl-CoA may be cleaved by beta-ketothiolase to form 2-acetyl-CoA, which then enters TC cycle and produce ATP. Thank you very much.